happy to be able to talk about the experience that we had with mating disruption for mealybugs in California. Okay, so in California, the principal vineyard mealybugs that we have are grape mealybug and vine mealybug. In Washington, of course, you have only the only the great mealybug. Luckily, you do not have vine mealybug yet, and hopefully that will be the case for a long time. Great mealybug is native to North America. It's a somewhat sporadic pest that generally has pretty small populations. We can get a lot of males, but the damage to the fruit is not usually great or excessive. In fact, they can be almost fairly difficult to find. And you can see that cluster that's cut apart there. In the entire cluster, those were the only 3 mealybugs that I found. Of course, great mealybug is a vector of grapevine leaf roll associated virus 3. And that, of course, has elevated the concern around mealybugs as pests in general, but great mealybug in particular. So just to make sure we're all on the same page about mealybugs and their life cycle. So the mature female, once she has mated, will lay eggs into an ovisac, which is what you see there. I'm just going to get my laser pointer ready. So here's the waxy ovisac, and the eggs are laid inside that ovisac. That protects them, keeps the humidity nice until they're ready to hatch. When they hatch, the first instar that hatches out of the egg is commonly called a crawler. And the reason why it's called a crawler is because it moves a lot and it moves fairly quickly. That's because they're very small and they're sensitive to dehydration. So they need to move away from the ovisac, settle down, and start feeding. Once they really do that, they'll tend to molt. And then you have 2 other instar stages, the second and third instars, before you get either an adult female or a male. Looking at the photos and the diagrams, you can probably tell that the male is the only life stage that has wings. But as a young instar or as a juvenile, it's impossible to tell the difference between females and males. So they all have the same rough body shape as juveniles. It's only once they're an adult that you can say, yes, that's a male mealybug there. Okay, so today we're going to be talking about mating disruption, and that is based around pheromones. Insects communicate via chemicals, and these chemicals are called pheromones. They are secreted by the through the exocrine glands by one individual, and then another individual of the same species receives that pheromone, and the pheromone elicits a specific response. It's really important that same species, right? Because if you were trying to communicate something about alarm or time for mating, you would want to be trying to attract the same species and not a different species. So there's all different types of pheromones. Aggregation pheromone may be one that you're familiar with simply because things like brown marmorated stink bug and spotted lanternfly that are in the press quite a bit produce aggregation pheromones. And so you'll see massive numbers of insects here in brown marmorated stink bug on a street tree in Sacramento. On the East Coast in Pennsylvania is where we get a lot of these photographs of spotted lanternfly aggregating on trees or buildings or other places. So the reason why insects aggregate is for security from predation. Sometimes they're trying to call their same species to utilize a scarce food resource. And it also results in cohesion of social insects who may be nesting together. Another type of pheromone is a spacing pheromone. The female moth that you see in this photo here, she's laying her eggs and then she'll cover them with a spacing pheromone. And then when another female moth of the same species goes up to that plant, she can detect that spacing pheromone and then knows that there are already eggs on that plant. So it allows that species to take good use of the resource and not to overpopulate a resource, because if there's too many eggs, there won't be enough food for the larvae when they hatch. Anyone who's ever watched ants or had ants invade their house has probably seen a trail pheromone. There have been over the years some trail pheromones that have been synthetically produced in order to increase recruitment to toxic baits. Alarm pheromones are also produced by insects to respond to the threat of a predator or some other kind of threat to the nest. Sex pheromones, which are used in attraction and courtship, are probably the most common pheromones that we use synthetically in IPM programs. And we use them both for detection and for mating disruption. And the process by which we use these is laid out here. So the first thing we need to do is we need to identify these compounds that are excreted by the female. And we usually do that using gas chromatography. So you see that coming off of the GC column here. This is where the pheromone came off. And then if you expose that to the male antennae, you can see that the male antennae are having a response only to this particular chemical that has been excreted or is coming off the column. So this is the structure of the grape mealybug pheromone, and then this is the structure of the vine mealybug pheromone. So in each of these cases where we're looking at using pheromones either for detection or mating disruption, we first go through the process of identifying these compounds and then make sure that they elicit the appropriate attennal response from the male. The next step is to determine whether we can synthesize those or not, and which biosynthetic pathway we might want to use for synthesis and their feasibility for mass production. Obviously, they're chemicals, so there's different levels of uh, complexity in those chemicals. So some may lend themselves more easily to mass production than others. And then lastly, once we've figured out if and how we can mass produce these pheromones, we want to try the synthetic versions of the pheromones in efficacy trials. So usually done in situ, so in this case in the vineyard, although you could also do them in a colony in a laboratory. So you want to make sure that if you're loading uh, a lure with the synthetic pheromone into a trap, and then you place that trap in the field, that it is then capturing males in the trap. Okay, so the synthetic pheromones that we use for male mealybug detection, we have used them for vine mealybug since about 2001 when that pheromone was identified. And then it was 2007 when the grape mealybug pheromone was identified. And the process for using them for detection is that the synthetic version of the pheromone is loaded into a lure. That lure is placed into a trap. The trap is placed in the field and then left in the field for a certain interval before it's collected. And then the mealybugs are identified and counted on the trap. So luckily for us, male mealybugs have a very distinctive color. So they're that sort of orangish red that you're seeing. And they also have a fairly distinctive body shape. So while there may be other insects in the trap that are the same size, between the color and the shape of the body, it's fairly easy to identify the males in the trap. So these are some zoomed in photos of the males that we usually count the traps using a microscope or a hand lens. And then if you were to just look at the trap in the field, this is what you would see. So each of these little orangish reddish specks that you're seeing is a mealybug male. And then here's a slightly more zoomed in view. So once you're trained, it is fairly easy and fairly quick to count the number of male mealybugs in these pheromone-baited traps. And the types of information that you get from them are shown in this figure here. So these are data from Washington, from some vineyard monitoring that was going on for great mealybug. And you can see that in the 2 years that were monitored, in sort of late May, early June is when the male started to be detected. In 2010, there were many more males detected in that first flight than in 2011. And in 2010, the first flight peaked slightly earlier than it did in 2011. Needless to say, there's 2 flights of the great mealybug males in both Washington and California. And that first one in Washington is in the June to July period. The second one starts in August and goes through October. And so information like this we can use to look at populations of great mealybug males over time. And then also within a single growing season, you can look at it and say, okay, well, here's when my flight's happening, and then here's when I might have nymphs present, and then you can use that in order to make decisions about interventions, such as insecticide sprays that might be going on. So that's the detection piece. And then the next piece is mating disruption. So similarly for detection, we take a synthetic version of the sex pheromone, but then it's loaded into a passive dispersal device, which I call a dispenser. So here's a couple of the dispenser designs that are available for vine mealybug. It can also be formulated as an aerosol, in which case it would go into a container, and this is the dispenser for the aerosol. And then also the vine mealybug main disruption product, it has been formulated as a flowable product. So you can make a spray application of that. And what you're seeing in the photograph here is a male mealybug mating with a female. And so the principles behind mating disruption, as I said, the female is releasing the pheromone. The male is using that pheromone plume in order to orient to the female for mating. In the case of when we put dispensers or we use some flowable or other type of product for mating disruption, we get clouds of pheromone within the vineyard. And that makes it very difficult for the male to orient to the female. And when the male can't orient to the female, that will either delay mating or it will mean that mating will not happen altogether. And that affects the reproductive output and efficiency of the mealybugs, meaning that fewer eggs are laid and those resources that would have been used for eggs or for reproduction are instead invested in survival. So the older the mealybug is, when she's mated, then the fewer eggs she's able to produce. So it has an effect of delaying mating in that you get less output of viable eggs. Okay, so the title of the talk, Mating Disruption for Vine Mealybug, it does work. So what we've seen with looking at different formulations, different dispensers over the years, we see both reduction in trap captures where we're using mating disruption and also a decreased damage to the fruit that's harvested. So on the left here, you're seeing that in the mating disruption plot, we are catching essentially no males. There's a little blip here. Probably that's because that's the time of year when the flight is peaking of that second flight. And so we do catch a few males in the mating disruption treatment, but significantly less than what we find in a control treatment. And then, as I said, we see reductions in population and reductions in damage to the bunches. And there was an article that was published in 2021 suggesting that mating disruption was applied across about 325,000 acres in that growing season. And it's been used in Europe, South Africa, South America, and also North America and California. This is for vine mealybugs specifically. And it has been a very important part of the integrated pest management program that we have for vine mealybug. Vine mealybug has very large populations and they can be difficult to control because they're often in protected locations under the bark. Like you're seeing here, they congregate in massive numbers, and then they can be protected by ants. And so um, we we use an IPM program in order to be successful with our vine mealybug management. That includes mating disruption, biological control, the judicious use of insecticides, and then suppression of ant population so that our biological control agents, particularly the parasitoids, can have access to mealybugs. So it's been a very important part of the IPM program for a number of years now for vine mealybug. If you're still questioning whether or not that pheromone is attractive to the males, Kent filmed this video here. Hopefully you can see it. This is a male mealybug in a petri dish. And that gray thing you're seeing is in fact not a mealybug female. It is a piece of the lure, which is what we use in the traps. So the male mealybug is trying to mate with that piece of the lure. So it is in fact the the chemical that we have that we use in the synthetic version is quite attractive to the males. So from dispensers, you'll see a slow release of the pheromone over time. That's the release there from the dispensers in Sicily and Venice in 2016 and 2017. We're not usually doing that outside of research trials, but if you're wanting to make sure that the dispensers are still active, that they're still releasing the pheromone, what we do is we use traps as a proxy for that. So we'll deploy a trap or two in the block that has mating disruption and just make sure that we're not catching any males in that trap, because if the males are not able to locate the trap, then that probably means that the dispensers are still actively releasing enough pheromone into the air to prevent or delay mating. So we do still use traps. Many people think that once we're using mating disruption that we don't use traps anymore, but we do still use them mainly just as a kind of a check and making sure that everything's still working okay with our dispensers or our applications. So the timing of deployment of the mating disruption should coincide with the male flights. So as we talked about for great mealybug in Washington, you would want to make sure that your dispensers are up by June, because that's when the males are going to start flying, and then they should be releasing, or you should have longevity of the dispensers through October. If a single application doesn't cover that period, then the manufacturer would probably recommend that there be two applications so that you can make sure that the entire periods of both flights are covered. Since I haven't worked with the Great Mealybug pheromone product, I'm not sure about dispenser longevity, but that's something that the manufacturers can talk to you about, or that you might hear from in the subsequent presentation. So in Napa anyway, our flight of great mealybug actually starts earlier, so we would want to make sure if we were using mating disruption for great mealybug that we had our dispensers up in April, and by late October is pretty much when we're no longer detecting males anymore. So a lot of people say, when do you put the dispensers up? And you need to know basically when the males are flying, and then what the longevity is of the whatever product you're going to be using. Another important thing about mating disruption is that continuity of use is very important. So this is not something where you use it one season and not the next season, then back to it the following season. So you get greater results and continual decreases in mealybug populations with consecutive use. So what we're seeing here is that these are blocks that started to have mating disruption in 2006, and we were still getting some damage to the fruit in 2006, but by the third year we were getting very little damage to the fruit, particularly in this block where we were finding very few vine mealybugs in the clusters. There were still some here, but you can see that we're getting increasing gains over time. So it's something that you're going to want to keep using consecutively to get the most out of the of mating disruption. That is also seen in this graph where the dark bars here are the first year that mating disruption was used, and then those certainly lighter bars are the second year. And so you can see, particularly with the high populations of mealybugs, it can take a couple of years before the populations really start reducing. And in fact, mating disruption is more effective at lower mealybug population density. And the reason for that is because mealybugs have a very clump distribution. So meaning a lot of them may be on a single vine, and then it may be a few vines in that area, and then it's the distance before you see any more. So in a clump distribution, and when they have very large populations, the male might not even leave the vine in order to mate. And so if it doesn't leave the vine, it may not rely on the pheromone at all to orient towards the female. And so that's why we say that the lower mealybug densities are where you see more effective mating disruption. So I have a little Where's Waldo for you. There is actually a male mealybug in this photograph. So there's a male mealybug there in among all of these females. So again, probably not even going to disperse, and it could mate with at least 10 females just, within that area. Other thing about mating disruption is that regional programs are very effective. So you can still be effective on a single block level, but when we have large regions that are using mating disruption, where the neighbors come together and decide that mealybugs are a problem. Excuse me, my allergies have not been great. So when the neighbors come together, because mealybug is a regional problem, we can see that when they're all using mating disruption, we have reduced mealybug density, and it also reduces spread. So what you're seeing here are the hotspots of vine mealybug in 2011 and then in 2012. And so you can see that there's not much change in 2011 or early in 2012, and it's not until later in 2012 that you start seeing some of those hotspots disperse. So we're just with increased use over time and across a larger area, we're seeing reductions in density and then also the risk of spread of the mealybug. The other interesting thing that we've observed with mating disruption, particularly with vine mealybug, is that there's a synergism with biological control. Specifically, this has been studied with the parasitoid Anagyrus vladimiri, which is the principal biological control agent of vine mealybug around the world. And that is the female Anagyrus that you're seeing photographed there. The female of the wasp lays her eggs into the mealybug, and the wasp larvae eats and consumes the mealybug from the inside out and kills it that way. Well, what we think is that the female wasp also orients to the mealybug pheromone because we see increases of biological control in blocks where we have mating disruption deployed. And what you can see from this study is that they did find anagyrus wasps in the traps, and in traps that were baited with the pheromone, they found more than they found in an unbaited trap. So we think that that synergism is really important, and that the mating disruption is somehow influencing the parasitoid search behavior. It's cueing the female to say that her host is in the neighborhood, and then keeping her there and keeping her searching in order to lay eggs into those host mealybugs. Sort of another thing about mating disruption is practice is thinking about wind direction and then also slope, and how that is going to influence the movement of the mating disruption particles, or the pheromone particles over time. So they could sink down a slope. And so if you look into the mating disruption literature for moth pests in particular, there are recommendations and guidelines around the number of dispensers that you might need at the top of the slope versus at the bottom of the slope, or in an upwind versus a downwind location. So just something to think about if you're seeing um, some issues with efficacy. You could start looking in, okay, am I on a slope, or am I in an upwind position? And what can I do to maybe increase the amount of pheromone particles that are in the air based on the dispenser densities? The other thing to know, and we don't really do this in practice, although if you had an aerosol dispenser, you could time it to releasing at certain times of the day. But we do know that temperature influences male flight activity. So beyond a certain temperature, the males are just not going to take off and fly. And in the laboratory, we can see that for vine mealybug, we have flights between the temperatures of 20 and 28 degrees Celsius, but above that, you don't get much flight activity, nor do you get much flight activity below that. Taking that out into the vineyard in the central part of California, so down in the Fresno area, it does get, very warm, definitely above 28 degrees Celsius during the day. And so what we see when we put traps out and count them on an hourly basis is that the male flight happens in the morning, and it is in response to those temperatures. So I've had people say to me, with the second flight of the great mealybug, it can often have many smaller peaks rather than one large peak. And I think part of that is an influence of temperature and the effect of temperature and what it has on the male flights. So it's just something to think about. It's not so much put into practice in mating disruption programs. Again, aside from, I suppose, if you had an aerosol dispenser and you could program it and to release only in a certain temperature range. So the other thing to remember about male mealybugs is that we do see natural interannual fluctuation in their populations and in trap capture. So without any mating disruption, sometimes with insecticides, other times with no insecticides, we do see fluctuations in populations through the years, comparing one season to the next. And we think that it's related to environmental conditions. Some of it may be around overwintering conditions, and then other may be related to the in-season conditions. So there are definitely fluctuations in mealybug populations, and you can see that in the trap captures in all the long-term trap capture data for great mealybugs. So lastly, I'd just like to touch really briefly here on leaf roll disease and mating disruption. So unfortunately, there's not a lot of literature or studies really around mating disruption and the role it could play in spread of leaf roll disease and reducing the spread of leaf roll disease. I think mainly because a lot of the field studies around leaf roll disease were conducted in New Zealand. They were conducted mostly with Citrophilus mealybug. There's mating disruption product for Citrophilus mealybug. And then in Napa, when we were studying leaf roll disease, we were mainly dealing with great mealybug, and we did not have an option for mating disruption at that time. So hopefully, if mating disruption were something that were used in an IPM program for mealybugs, either vine mealybug or great mealybug, that reduction in populations and spread would also be reflected in the incidence of leaf roll disease over time. So that may be something that in Washington you have an opportunity to study and work on, because of the opportunity of a commercial mating disruption product for great mealybugs. So we don't have a lot of really detailed information around specific or really around mating disruption for mealybugs and then leaf roll disease. So with that, I will wrap up my presentation, say thank you very much for your attention, and to my funding sources and my collaborators. And everyone.
Thank you, Monica.